السلام عليكم ورحمة الله وبركاته. الحمد لله رب العالمين، والصلاة والسلام على أشرف الأنبياء والمرسلين سيدنا محمد وعلى آله وصحبه أجمعين. This is a video recording for the respiratory physiology lectures on pulmonary ventilation in the respiratory block. These are the intended learning outcomes for these learning activities, and these intended learning outcomes are very important and we need to focus on them because, as you know, this is a sort of the contract between the instructor and the students. These intended learning outcomes, they are clearly defined statements which begin with action verbs, like, for example, define breathing terms, state functions of the respiratory system, describe, differentiate, describe, explain. These action verbs determine the level of the learning which is expected from you after you have reviewed the PowerPoint presentation and the associated learning material. Here we have the recommended learning resources for these learning activities. You need to listen to these video recordings before coming to the lecture, and you may need to repeat some parts of these video recordings several times in order to understand the explained concepts and principles, because I'm not going to repeat the entire thing again during the lecture time. The lecture time would be allocated to the application of the knowledge which you must have gained from listening to the video recordings. So now let's begin these learning activities by defining breathing terms. And the first term is eupnea. Eupnea means normal, quiet breathing at rest. When we breathe approximately 12 to 20 breaths per minute, and in each breathing cycle approximately 500 milliliters of air, which is equivalent to half liter of air, gets in and half liter of air gets out. Tachypnea, tachy means fast, so tachypnea means an increased respiratory rate greater than 20 breathing cycles in 1 minute, and in tachypnea breathing is typically shallow. Hyperrhythmia, this means hyper means increased, but in this case what is increased is the ventilation in order to meet the metabolic requirements of the body. So ventilation is increased in hyperrhythmia to satisfy the metabolic needs of the body, because the body in some conditions needs more oxygen and generates more carbon dioxide. So in order to provide the body with more oxygen and to eliminate the more carbon dioxide which is generated, the body should have hyperrhythmia. So in hyperrhythmia we have either increased respiratory rate, which is tachypnea, and or overly deep breaths. So in this case, in hyperrhythmia, we could have either increased respiratory rate higher than the 20 breaths per minute, and or overly deep breaths. And this happens, for example, during muscular exercise and during the respiratory compensation to some conditions in the body, for example, when there is a metabolic acidosis in the body. Now, hyperventilate is another breathing term which means increased ventilation. So what's then the difference between hyperrhythmia and hyperventilate or hyperventilate? The difference between hyperapnea and hyperventilate is that hyperapnea is increased ventilation to satisfy, to meet the metabolic requirements of the body, whereas hyperventilate, it is also increased ventilation, but now the increased ventilation has exceeded the metabolic requirements of the body. Also in hyperventilate, there is increased respiratory rate, there is tachypnea, where the breathing rate is higher than 20 breaths in 1 minute, and there is also and or overly deep breaths. Hyperventilate is seen in cases of an anxiety attack, when someone is under an anxiety. Bradyapnea, brady means slow, is the opposite of tachypnea. So this is decreased respiratory rate, less than the lower rate of breathing, the lower rate of respiration, so less than 12 breaths in 1 minute. Hypopnea is the opposite of hyperapnea. So this is decreased ventilation, also in order to meet the metabolic requirements of the body. So in some conditions and in some states of the body, the metabolism is decreased. So in this case, there is no need to have the eupnic state. So in this case, in hypopnea, for example, when someone is sleeping, he doesn't need much oxygen as when someone is awake, or for example during the respiratory compensation of some conditions. So hypopnea is then decreased respiratory rate, predypnea, and or shallow breaths. So it is the opposite of hyperapnea. Now hypoventilate, there is also decreased ventilation. But what is then the difference between hypoventilate and hypopnea? In hypoventilate, the decreased ventilation is abnormal, is not normal, and it is associated with abnormalities. And there is also decreased respiratory rate, which is predypnea and or shallow breaths. And this is seen in abnormal conditions like pulmonary diseases and hypoventilation syndrome. Dyspnea, the P is not pronounced, so dyspnea is labored breathing, and this happens during strenuous exercise or when there is pulmonary embolism or when there is congestive heart failure. Apnea means cessation of breathing, stoppage of breathing, like with sleep apnea. So we have finished then with these breathing terms, and let's very briefly go through the importance of respiration. Why do we need to have the respiratory system in the body? You all know that oxygen is very important for our survival. We need oxygen to generate most of the energy. I'm saying most, I'm not saying all the energy, because the body can generate oxygen even in the absence of oxygen during anaerobic metabolism. But in the presence of oxygen, most of the energy required for our cells to function properly is generated. So we call the process or the processes of supplying oxygen to the cells of our body and removing carbon dioxide respiration. So respiration is the entire story, the entire scenario of providing the cells of the body with oxygen and then removing carbon dioxide from the body and eliminated to the atmosphere. During rest, in normal adult male takes up approximately 250 milliliters of oxygen. So the resting adult male, the cells of the resting adult male require 250 milliliters of oxygen to generate the energy required for them to function appropriately, and gives off 200 milliliters of carbon dioxide in each minute. So here we have the equation which shows the aerobic production of energy. So the food which we eat, the carbohydrates, the lipids which we eat, they are digested and then absorbed, and they go to the tissues, and in the tissues, when the food is oxidized, we require oxygen. So oxygen is required, and oxidation of food results in the production of usable energy for the cells in the form of adenosine triphosphate. And in this process of energy production, carbon dioxide and water are produced, and carbon dioxide is not required for the body and needs to be eliminated from the body. So this table summarizes the functions of the respiratory system. So the major and the most important functions of the respiratory system are to provide the cells of the body with oxygen and to eliminate carbon dioxide. But the respiratory system has got other important functions. For example, regulation of the hydrogen ion concentrations in the fluids of the body, in the plasma and in the interstitial fluid. And this is done in conjunction with the kidneys. Also, the respiratory system is important for phonation, for speech. It is important for defense against microbes. And it also influences the concentrations of important chemical messengers in the blood. So when the blood moves through the pulmonary circulation, through the pulmonary capillaries, some of the chemical messengers can be removed from the pulmonary capillary blood, and some other chemical mediators, messengers, can be added to the blood as it passes through the pulmonary capillaries. Another important function is, as blood moves through the pulmonary circulation, this blood might contain small clots, and the importance of the blood moving through the pulmonary capillaries in this case is that the small clots, they are trapped and dissolved, okay, and they do not reach the left side of the heart. So if we sit for a long time, or if we sleep for a long time, so people who are bedridden and who sit for a long time, they might develop some clots in the deep veins of their legs, and these clots might move, and they go to the right atrium, then they go to the right ventricle, and then they move along the blood into the pulmonary trunk and the right and left pulmonary arteries. But while moving through the pulmonary capillaries, they get trapped and dissolved. Now, before discussing the pulmonary ventilation in details, we need to very briefly describe the functional anatomy of the respiratory system. So what is the functional anatomy of the respiratory system, or what are the functional components of the respiratory system? The respiratory system is functionally divided into the thorax, which is also known as the chest cage, the respiratory muscles, and the lungs and the airways. And we will describe these functional components very briefly one by one. So what is the thorax or the chest cage? The thorax or the chest cage is formed by the rib cage, these are the ribs, and the sternum anteriorly, and the thoracic vertebrae posteriorly. Now this chest or the chest cage is not a rigid structure, rather it is an elastic structure, and the elasticity of the chest cage is mainly due to the joints between the ribs with the sternum anteriorly, and the joints between the ribs with the thoracic vertebrae, with the sternum anteriorly and with the thoracic vertebrae posteriorly. As you can see here, the ribs, they are torqued. Torqued, it means twisted. It is as if you are bending something to fit between these two attached points. So this rib is bent, so that is now it is attached here anteriorly and attached here posteriorly. And because this rib is bent, torqued, twisted, so it tends to move out. So this leads to expansion force on the thorax. So the chest cage, it tends to move out, okay, because the ribs, they need to resist this twisting and their attachment between the sternum anteriorly and the thoracic vertebrae posteriorly. Now let's move on and describe the muscles of breathing. The muscles of breathing or the muscles of respiration, they are divided into inspiratory muscles, muscles for inspiration, and expiratory muscles, muscles for expiration. The inspiratory muscles, they contract in order to expand the thoracic cavity in all dimensions, in the vertical dimension and in the transverse dimension and also in the anteroposterior dimension. And when the thoracic cavity expands, this leads to inspiration and air gets in. Accordingly, inspiration is an active process because it requires the inspiratory muscles to contract first, then the thoracic cavity expands, and then air gets in. It is very important to realize that the chest cavity expands first and then air gets in. It is not that the chest cavity expands because air gets in. In fact, air gets in after the chest cavity has expanded as a result of contraction of the muscles of inspiration, and we will describe the dynamics of inspiration and expiration in details during this video recording. Then, after inspiration is over and air has entered into the two lungs, the inspiratory muscles, they relax and they bring the thoracic cavity back to its resting volume, and this leads to expiration. So normal resting quiet expiration does not require muscles to contract. Rather, what happens during normal resting expiration, the inspiratory muscles relax. Hence, normal resting expiration is a passive process. The main inspiratory muscles include the diaphragm, which is a dome-shaped muscle, and when it contracts, it flattened, and this expands the chest cavity in the vertical dimension. And we have got muscles here between the ribs known as the external intercostal muscles or external intercostals, and when these muscles contract, they move the chest cage up and out. So they expand the thoracic cavity in the transverse dimension and in the anteroposterior dimension. These are the main inspiratory muscles, the diaphragm and the external intercostals. We've got accessory inspiratory muscles, which include pectoralis minor muscle, and this is not used during normal resting inspiration. Rather, it is used during forced or labored inspiration. It is a muscle here. We have got here muscles in the neck known as the sternocleidomastoids. One sternocleidomastoid on the right side and then another sternocleidomastoid muscle on the left side, and also these muscles are not used during normal resting quiet inspiration. Rather, they are used during forced or labored inspiration. And we have got here also muscles in the neck known as the sclene. They are also used during forced or labored inspiration. Now, the expiratory muscles, they are not used during normal resting expiration. We do not need muscles to contract during normal resting expiration when air gets in, when air gets out while we are resting. All what happens is that the inspiratory muscles relax. The inspiratory muscle relax. So why then do we need to have muscles for expiration? We have these muscles in order to use during forced or labored expiration. And when expiration is forced or labored, and we need to have the muscles of expiration contracting, in this case, expiration becomes an active process, and these expiratory muscles include the muscles of the anterior abdominal wall, which are the rectus abdominis. Here we have another rectus abdominis, and the oblique muscles. We have the external oblique and we have the internal oblique, and also here we have the internal intercostal muscles. So again, Here we have, so this is the scenario at rest. This is the chest cage, the thoracic cavity at rest. So what happens now during inhalation or inspiration? The diaphragm contracts and it moves down, expanding the thoracic cage, the thoracic cavity in the vertical dimension. The external intercostals contract, expanding the thoracic cavity in the anteroposterior dimension and in the transverse dimension. So these are the muscles which are used during normal, quiet, resting inspiration. If inspiration becomes labored and forced, then we need to have contraction of the accessory muscles of inspiration, which lift the thoracic cage further up. And these accessory muscles of inspiration include the pectoralis minor, the spleens, and the sternocleidomastoids. The exhalation at rest is a passive process. We do not require muscles to contract. All what happens is that the muscles of inspiration relax, but when expiration becomes labored and forced, we need to have contraction of the muscles of the anterior abdominal wall and the internal intercostal muscles. So now we have finished with the description of the muscles of breathing, and let's move on and describe another functional component of the respiratory system, which are the lungs. The lungs are located within the thoracic cavity, but in separate pleural sac. So each lung is surrounded by a pleural sac, is not directly attached with the thoracic cage. The lungs are elastic structures. They look like balloons, two balloons. And you know, a balloon is an elastic structure. When you inflate a balloon, it allows you to inflate it because the wall of the balloon is elastic. It can be stretched. However, at the same time as the balloon is inflated, these elastic fibers which we have in the wall of the balloon, they become stretched and they develop a recoil force inwards. Same thing applies to our lungs. They are elastic structures. In fact, they are much elastic and they are much compliant than a balloon. So they are stretchable, and when air gets in, they expand because they have elastic fibers, or we call elastin fibers, in the interstitium. And the interstitium is the tissue and the space which surrounds the alveoli and also in the walls of the alveoli. So these elastic fibers, they allow the lungs to be stretchable, but as they are stretched like a rubber, there is an inward recoil force. Now, under normal conditions, our lungs are always being stretched, even after expiration. So when air has gone out of our lungs, this doesn't mean that all the air in the lungs has gone out. Still, there is a volume of air which remains in the two lungs, and this volume is still expanding the two lungs. And since the two lungs are still expanding even at the end of expiration, so they are stretched, and so the lungs are in a constant state of recoil. It is like a balloon. But when we deflate a balloon, all air gets out. But in our lungs, during expiration, not all air gets out. Some air remains in the two lungs, as we will describe in this video recording. So here we have, so these are the two lungs, and the anatomical description of the two lungs will be described in anatomy. So this is the right lung, which is divided into three lobes: the superior lobe, the middle lobe, and the inferior lobe. And this is the left lung. We have the superior lobe and the inferior lobe, and this is the chest cage or the thoracic cage. Now, we've mentioned that the lungs are located in the thoracic cavity, but in separate pleural sacs or pleural compartments, as we have just mentioned. So what are these pleural sacs or pleural compartments? So this is a pleural sac, and embryologically, developmentally, each one of us during the intrauterine life, while we are still in the uterus of our mothers, we have got two pleural sacs. One pleural sac on the left side and the other pleural sac on the right side. And the lung develops by pushing into the pleural sac, like this hand pushing into this fluid-filled balloon. So what happens then? Imagine as this is the lung, and this is the pleural sac. What happens then as this lung develops and pushes into this fluid-filled balloon? Now this is the lung, developing lung. The pleural sac now takes the shape of the lung, and it is no more a balloon. Rather, it will be a double-layered sac like this. This is a layer and this is another layer. So now the pleura becomes a double-layered serous. Serous means that it is filled with a thin fluid layer, membranous sac, which has got now two layers and is taking the shape of the lung. One layer this is now the lung, and these are the two layers of the pleura. One layer of the pleura is lining the thoracic cavity and attached to the diaphragm, and this layer of the pleura is known as the parietal pleura, and the other layer of the pleura covers the lungs, and this is known as the visceral pleura. And what do we have between the parietal pleura and the visceral pleura? There is now a fluid filled potential space, very thin potential space, which is filled with a thin fluid layer between the parietal and the visceral pleura. Now the pressure in this pleural sac, it is subatmospheric pressure. It is below the atmospheric pressure. You know, the pressure in the atmosphere, this is the pressure which we are all exposed to, and this is equivalent to 760 millimeters of mercury. If we have got a container of mercury and we have got an inverted tube, mercury will elevate in this inverted tube 760 millimeters of mercury because of the pressure of the atmosphere, because of the weight of the atmosphere, and this is what we call the atmospheric pressure, or simply we call it zero pressure. Now the pressure in the pleural sac is subatmospheric. Why? Remember earlier on I've mentioned that the chest cage, it is an elastic structure, and because the ribs are torqued, they are twisted, they tend to move out. So the chest cage tends to move out, and the lung tend to recoil in. So now we have got two forces. This force of the recoil of the chest cage out, and this is the recoil of the lung in. So now the pleural sac is exposed to these two opposing forces which are moving away from each other, so they tend to expand the pleural sac, so the pressure decreases. This is a physical aspect. So structure of the pleura allows then the lungs to move along with the thorax, because the parietal pleura is attached to the diaphragm and also lines the chest cage, and this is the visceral pleura. So if the chest cage expands, the lungs will also expand. Still air has not gone in yet. So this is another figure which shows the pleural sac. So this is our two lungs. This is a transverse section. So the layer which is red in color, this is the visceral pleura, and the layer which is blue in color, this is the parietal pleura. And remember, we have got two separate pleural sacs, and we do not have air inside the pleural sac. Rather, we have a thin fluid layer in the pleural sac. Here we have another picture which shows, so this is the lung, this is, we have opened the chest cavity, so this is a rib, these are the intercostal muscles, this is the lung, and this is the parietal pleura which lines the thoracic cavity, and this is the visceral pleura which covers the lung, and in between these two layers is the pleural sac with thin fluid layer. So this is what I've just mentioned. So the visceral pleura is separated from the parietal pleura by a very thin fluid layer, approximately 10 micrometer thick, and a micrometer is 1 part of a million part of a meter. So if I've got a meter and we divide this meter into million parts, each small part is a micrometer. So now we're finished with the description of the thoracic cage, the muscles of breathing, the lungs, the pleural sacs, and now let's move on and describe the airways very quickly. Functionally, the airways or the air passages are divided into 2 zones. What are these zones? One zone is known as the conduction zone or the conducting zone, and the other zone is known as the respiratory zone. The conducting zone, it expands from the nearest, from the opening of the nose down to the terminal bronchioles. Or some people, they prefer to start the conducting zone from the trachea, and they give the trachea the level 0. So from zone 0, which is the trachea, up to zone 16, which is the terminal bronchioles, this is the conducting zone. So zone 0 starts at the trachea. Trachea is level 0 or zone 0. Now the trachea branches into 2 main bronchi, and the 2 main bronchi branch into lobar bronchi, which go to the loops, and the lobar bronchi divide into segmental bronchi, which go to the segments, to the bronchopulmonary segments, and the segmental bronchi divide into subsegmental bronchi, and so on. And then the bronchi divide to give rise to bronchioles. The bronchioles divide to give smaller bronchioles, and then we have got the terminal bronchioles. After the terminal bronchioles, we have got the respiratory bronchioles, where now alveoli start to appear in the respiratory bronchioles. Then the respiratory bronchioles keep on dividing and give the alveolar sacs branching. So now from zone 0 up to zone 16, this is the conducting zone. Zone 0 is the trachea, zone 16 the terminal bronchioles, zone 17 is the beginning of the respiratory bronchioles, up to zone 23. Now this numerical designation, the 0, the 1, the 2, the 16, within each of the zones indicates the degree of branching. So the trachea is 0. Why? Because there is no branching in the trachea. The trachea, it branches at level 1. Okay, zone 16, it means that there is 16 levels of branching, and so on. So here we have, so this is our trachea, zone 0. Then this is the main bronchi, level 1. Lobar bronchi, level 2. Segmental bronchi, level 3. Even 4, 6, 5, 6, 7. They are all bronchi. Then here we have got bronchioles, and up to the 16th level, this is terminal bronchioles. Then respiratory bronchioles begin at level 17, keep on up to level 21, where the alveolar ducts begin. Then alveolar duct also branch 22. Then we have got the alveolar sacs 23. So alveoli, these are the terminal bronchioles here. Level 16, there are no alveoli. Alveoli start to appear in the respiratory bronchioles at level 17, and they keep on. These are the alveolar sacs, and these are the alveoli. Now, air, as it flows along the respiratory passages or the airways, it meets resistance. As when blood flows through the blood vessels, it meets resistance because of the friction between air and the walls of the air passages. Now, the greatest resistance to the flow of air is met in zones 1 up to zone 7, which represent the large and the medium-sized bronchi or airways, as we will describe in the next learning activity. So we've got then, since there are no alveoli in the conducting zone, so there is no exchange of gases taking place in the conduction zone or in the conducting zone. Exchange of oxygen and carbon dioxide takes place in the respiratory zone, where we have alveoli. So then, why do we need to have a conducting zone? Are there functions for the conducting zone? There are no alveoli, so there is no exchange of gases. Yes, there is no exchange of gases, but there are other functions of the conducting zone. There are other functions for these airways. What are these functions? Now, the functions of the conducting zone controls airflow into and out of the respiratory zone. Because now, as air gets into the respiratory zone, the velocity of air has to be reduced, so that now time is allocated for the process of exchange of gases. So the entry of air and the exit of air from and into the respiratory zone is controlled by the conducting zone. Another function of the conducting zone is that as air moves along the conduction zone and before it reaches the respiratory zone, it is warmed and humidified. Sometimes we breathe cold air, and cold air is not good for the respiratory zone. So as air now moves along the conduction zone or the conducting zone, it gets warmed. Sometimes we breathe dry air, and dry air is irritant to the alveoli. It is not good for the alveoli. However, by the time it reaches the respiratory zone where we have the alveoli, air would have become saturated with water vapor and humidified. Also, it filters and protects the respiratory tract from the pollutants and the pathogens. We have hairs in the nose, and we have mucus, which trap large particles. Small particles escape trapping in the nose. So what happens to this? We might have bronchoconstriction. This is contraction of the smooth muscle cells of the airways when polluted air is inhaled. Now, fine particles which escape trapping by the hairs in the nose, they stick to the mucous membrane which lines the airways of the conducting zone, and then the cilia of the ciliated epithelial cells move and eliminate them from the air passages. Now, the movement of air through the airways of the conducting zone is by bulk flow, which means that the movement of air is by the pressure gradient between the atmospheric pressure and pressure within the respiratory system. The resistance to the flow of air through the conducting zone is controlled by the tone of the smooth muscle cells which we have in the walls of the airways of the conducting zone. Now, in these smooth muscle cells, we have got beta-2 adrenergic receptors, which when stimulated result in relaxation of the smooth muscle cells in the walls of the airways of the conducting zone, leading to bronchodilation or bronchodilation. And we've got also muscarinic acetylcholine receptors, which when stimulated by acetylcholine or cholinergic agonists result in contraction of the smooth muscle cells, leading to bronchoconstriction. Now, the beta-2 receptors which we have in the plasma membrane of the smooth muscle cells in the walls of the airways of the conducting zone are stimulated by adrenaline and noradrenaline, epinephrine and norepinephrine, and they are also stimulated by the drugs which act like adrenaline and noradrenaline, which we call them beta 2 agonists, like for example salbutamol or albuterol. And these drugs, because they cause relaxation of the smooth muscle cells and bronchodilation or bronchodilatation, they are used in the treatment of obstructive lung or obstructive pulmonary diseases when there is airway constriction. Now these beta 2 receptors, they are inhibited by beta 2 blockers, which we also call beta 2 antagonists. The muscarinic receptors, which we have in the plasma membrane of the smooth muscle cells in the airways of the conducting zone, they are stimulated by acetylcholine, and they are also stimulated by the muscarinic receptor agonists, the cholinergic drugs, and they are inhibited by the muscarinic antagonists, the muscarinic blockers or the anticholinergics, like for example ipratropium bromide. And when they are blocked, the result would be relaxation of the smooth muscle cells and bronchodilation and reduced mucus secretion, and hence these blockers, the muscarinic antagonists like ipratropium bromide, they are used in the treatment of obstructive pulmonary diseases like the beta 2 agonists. Now this figure summarizes what we have mentioned earlier about an important function of the conducting zone, which is the mucociliary escalator. What's the meaning of this mucociliary escalator? This figure shows the histological structure of the wall of an airway in the conducting zone. This is the real histological structure, and this is a drawing of the wall of an airway in the conducting zone. So here we have got cells which are known as goblet cells, and these goblet cells, they produce mucus, and this mucus traps the fine particles, including even the pathogens, which escape trapping by the hairs which we have in the nose. Now we have got also the pseudostratified ciliated epithelial cells, which have got cilia, and these ciliated cells, they are damaged by cigarette smoking, and hence cigarette smoking plays a role in infections and diseases of the respiratory system. And the mucus which is produced by the goblet cells is thickened in a disease known as cystic fibrosis, and this is a genetic disease which must have been described to you in the genetic course. And when this happens, now the cilia would not be able to propel this thickened mucus upwards in order to eliminate it from the respiratory system. Hence this thick mucus accumulates, and it is a good medium for the growth of the pathogens, and hence it causes frequent respiratory infections, and it also causes airway obstruction. Now we have finished with description of the functions of the conducting zone, and let's move on and describe the major functions of the respiratory zone. And we've mentioned that the respiratory zone begins from the level 17 respiratory bronchioles up to level 23, where we have the alveolar sacs and the alveoli. And in the respiratory zone, as you can see here in the figure, we have got alveoli. So this is respiratory bronchiole. This is an alveolar sac where we have alveoli, and these alveoli, they are surrounded by dense network of pulmonary capillaries. So the major function is thus the exchange of the respiratory gases. So oxygen in the alveolar air diffuses from the alveoli into the blood, and carbon dioxide, from the venous blood diffuses into the alveoli in order to get eliminated into the atmosphere. Thus, the primary function of the respiratory zone is the exchange of gases through the alveolar walls and the walls of the pulmonary capillaries. How many alveoli do we have in each lung? We have approximately 300 to 500 millions alveoli in each lung. So oxygen diffuses from air inside the alveoli, from the alveolar air into the blood in the pulmonary capillaries, and carbon dioxide diffuses in the opposite direction from the blood in the pulmonary capillaries into the alveolar air. Another function of the respiratory zone is protection, because we also have macrophages in the alveoli which engulf small particles which are even missed by the mucociliary escalator system. Now these alveoli, they are lined with a thin fluid layer, and on top of this thin fluid layer we have a thin layer of a material which is known as the surfactant. The surfactant is chemically lipoprotein produced by type 2 pneumocytes. These are cells which line the alveoli along with another type of cells known as type 1 pneumocytes. And the surfactant is very important because it reduces the surface tension forces which are generated at the interface between the thin fluid layer which lines the alveoli and air inside the alveoli, and this will be described in details in the subsequent learning activities. And it also contains proteins which are similar to some of the proteins of our complement system, which is a major component of our immune system. So here we have got, so these are the alveoli, these are the walls of the alveoli, as you can see. These are the pulmonary capillaries surrounding the alveoli. These are the pneumocytes which line the alveoli, and these are the macrophages within the alveoli. So here we have the major function of the respiratory zone, which is the exchange of gases at the alveoli. Oxygen diffuses from the alveolar air into the blood in the pulmonary capillaries, and carbon dioxide diffuses in the opposite direction from the blood in the pulmonary capillaries into the alveolar air in order to be eliminated to the atmosphere. Now, since the conducting zone does not participate in the process of respiration, it doesn't participate in the process of exchange of oxygen and carbon dioxide because there are no alveoli in the airways of the conducting zone. Some physiologists call the conducting zone as the dead space. It is dead only in terms of exchange of gases. So the anatomical dead space is represented by the conducting zone volume. So this is the volume of air inside the airways which does not participate in the process of exchange of gases. And as a rule of thumb, the volume of the anatomical dead space, the volume of the conducting zone in milliliters, is equivalent to the natural weight of the subject in pounds. So if someone weighs 200 pounds, then his anatomical dead space is approximately 200 milliliters. And the average value of the anatomical dead space is about 150 milliliters of air. So these 150 milliliters of air which fill up the conducting zone where we don't have alveoli, they do not participate in the process of exchange of gases. Can we have alveoli which do not participate in the process of gases? We can have this, and if this happens, these alveoli, or the air which is present inside these alveoli, constitutes what is known as the alveolar dead space. So alveoli which do not participate in the process of exchange of oxygen and carbon dioxide are known as alveolar dead space, and this could happen as a result of lack of blood supply or damage to the alveoli. So if these alveoli do not receive adequate blood, then they will not participate appropriately in the process of exchange of gases, and the air inside them will constitute alveolar dead space. In healthy subjects, healthy individuals, it is a very, very minimal volume. Now, if we combine the anatomical dead space together with the alveolar dead space, we come up with another dead space, which some physiologists call it the physiological dead space. So the physiological dead space is a combination of the anatomical dead space and alveolar dead space, and in normal healthy individuals, the physiological dead space is approximately equal to the anatomical dead space, because the alveolar dead space is very, very minimal in normal healthy individuals. So now this is the conducting zone. This is the air which we have inside the conducting zone where we do not have alveoli. So this constitutes the anatomical or the anatomic dead space. Now these alveoli, they are okay, they are well perfused, they receive adequate blood flow, but look at this alveolus. This alveolus is not receiving adequate blood flow. So if this is the case, then not all the air inside this alveolus would participate in the process of exchange of gases. Maybe part of the air will participate in the process of gases, but this part will not participate in the process of exchange of gases and will constitute what is known as the alveolar dead space. Now, we have finished with describing the basic concepts and principles of the respiratory system in man, and now let's very briefly describe the stages or the steps of respiration in man. In humans, respiration involves a series of integrated steps or stages or processes. It is not a single step process like what we have in unicellular organisms like amoeba. In amoeba, respiration is a single step process where oxygen diffuses from the environment into the amoeba cell, and then carbon dioxide diffuses in the opposite direction. But since humans are multicellular organisms, this cannot happen, and respiration involves a series of integrated processes which include, number one, pulmonary ventilation, which is the inflow and the outflow of air between the atmosphere and the alveoli. Then we have the exchange of gases, oxygen and carbon dioxide, between the alveolar air and blood in the pulmonary capillaries. Then we have transport of oxygen and carbon dioxide, and then we are going to have another exchange process of gases taking place at the level of the tissues, where oxygen moves from the systemic capillaries to the tissues, and carbon dioxide diffuses in the opposite direction from the cells of the tissues to the systemic venules. And oxygen, which is provided to the cells of the body, is utilized inside the cells by the mitochondria in a process known as internal respiration or cellular respiration. So respiration is then the entire process of supplying the cells of the body with oxygen and removing carbon dioxide. Respiration is not only pulmonary ventilation. Now, over the various lectures and learning activities of the respiratory block, we are going to describe these stages of respiration in details. So here we have got, this is the atmosphere, so the inflow of air bringing oxygen into the alveoli, and the outflow of air from the alveoli into the atmosphere taking off carbon dioxide to the atmosphere. This is pulmonary ventilation. Then the second stage of respiration is the exchange of gases. Then we have got transport of gases. Oxygen is transported by the arterial blood from the lungs to the left atrium, then to the ventricle, to the aorta, branch to the aorta, to the tissues of the body, to the cells of the body. Then you have got another exchange process taking place at the level of the tissues. The tissues take up oxygen from blood and give blood carbon dioxide. Now blood becomes venous blood or deoxygenated blood, returning back to the heart, to the right atrium, right ventricle, and then the right ventricle pumps this blood into the pulmonary capillaries. This diagram also summarizes the stages of respiration in man. I am not saying the stages of breathing, because breathing is just the first stage of respiration in man. Breathing is equivalent to pulmonary ventilation. Breathing which is inspiration and expiration, breathing which is inhalation and exhalation, is equivalent to pulmonary ventilation, which is the first stage of respiration in man. So this is pulmonary ventilation, inspiration, expiration. The Americans, they prefer to call inspiration inhalation and expiration exhalation. Then we have got the exchange of gases taking place at the level of the alveoli and pulmonary capillaries. Then I've got transport of gases, another exchange process taking place at the level of the tissues, and then oxygen is utilized by the mitochondria of the metabolizing tissues to produce cellular energy, and during this process carbon dioxide is produced and it has to be eliminated to the atmosphere. Now let's focus on the first stage of respiration in man, which is pulmonary ventilation, which is also known as breathing, which includes inspiration, expiration, inhalation, exhalation. Pulmonary ventilation, as we have just mentioned, is the inflow of air bringing oxygen and the outflow of air taking off carbon dioxide between the atmosphere and the alveolar air. So what's then the mechanism which is responsible for the movement of air between the atmosphere and the alveoli? We've mentioned earlier during this learning activity that air moves through the conducting zone of the airways by bulk flow. So we need then to have a difference in pressure between the atmosphere and between the alveolar air in order for air to move either in or either to move out. And we've mentioned that the diffusion of gases, the process of exchange of oxygen and carbon dioxide, takes place in the respiratory zone when the velocity of the flow of air is significantly reduced as a result of the huge cross-sectional area of the respiratory zone. So now airflow is a function of the pressure differences between the alveoli and the atmosphere. This is the pressure gradient, and it is inversely proportional to the resistance to the flow of air through the airways. Now air enters into the alveoli when the alveolar pressure is smaller than the atmospheric pressure, and air exits the lungs when the alveolar pressure is greater than the atmospheric pressure. Now, it is thus evident that we need to have a pressure gradient between the atmospheric pressure and the alveolar pressure. Now, the generation of this pressure gradient, it relies upon the important physiological relationship between the lungs and the chest wall and the pleural sac. The pleura plays a major role in the generation of the pressure gradient between the atmosphere and the alveoli, as we will describe. Now, since the atmospheric pressure cannot be changed, in order to generate a pressure gradient between the atmospheric pressure and the alveolar pressure, we need then to have a change in the alveolar pressure. How comes a pressure changes? There is a relationship between the volume and the pressure, and this relationship between the volume and the pressure is explained by the ideal gas law, or is explained by the Boyle's law, which states that an increase in the volume, when the volume of a space increases, the pressure inside this space decreases, and the opposite is true. Decrease in the volume increases the pressure. How do we measure the pressure? We measure the units which we use to measure the pressure are the centimeter of water or millimeter of mercury. And 1 centimeter of water is equivalent to 0.74 millimeters of mercury. So this is the ideal gas law. As you can see, the pressure is inversely proportional with the volume. As the volume increases, the pressure decreases, and as the volume decreases, the pressure increases, because we divide by the volume. So this is again Boyle's law, which states that the pressure of a fixed number of gas molecules is inversely proportional to the volume of the container in which this number of gas molecules are present. So now we have got this number of gas molecules inside this container. Now if we compress this container and we reduce the volume, what happens? The pressure increases. The opposite is true. If we decompress this container and we increase the volume, still the same number of gas molecules are present, but what happens to the pressure? The pressure decreases. So for a given quantity of a gas in a chamber, the pressure is inversely proportional to the volume of the container, and this is Boyle's law. Now we need to define some very important pressures which are important in the process of pulmonary ventilation or breathing. The first important pressure is the atmospheric pressure, or we call it the barometric pressure. This atmospheric pressure, or the barometric pressure, is equivalent to 760 millimeters of mercury, and it is because of the weight of the gases which we have in the atmosphere, because the gases in the atmosphere has got weight, and this weight exerts pressure on everything which lives on the surface of Earth. If we have got a container filled with mercury, and there is an inverted tube put on top of this container, the mercury will rise in this tube up to 760 millimeters because of the pressure of the air on the surface of the mercury container. For simplicity, we call the barometric pressure, or the atmospheric pressure, zero pressure. Its value is not zero, but we call it just zero pressure. Its name is zero pressure. Its value is 760 millimeters of mercury. Airway pressure, this is the pressure inside the airways. Alveolar pressure, PA, is also known as intra-alveolar pressure or pulmonary pressure. Now the pleural pressure, which you call it PPL, or intrapleural pressure, this is the pressure within the pleural sac. And under normal conditions, the pressure within the pleural sac is less than the atmospheric pressure. And since we call the atmospheric pressure 0, and the pleural pressure is less than the atmospheric pressure, which is 0, so the pleural pressure is then subatmospheric, or we call it a negative pressure. Now, if we have a space with a negative pressure, this space tends to shrink its volume in order to elevate its pressure based in Boyle's law. So, having said so, the pleural sac which surrounds the lungs, they tend to shrink because of the negative pressure inside them. So this is good for the lungs because the lungs are surrounded then by a space which tends to shrink, and this would then facilitate the process of lung expansion, as we will see. So the negativity of the intrapleural pressure is because of the two opposing forces acting on the pleural sac, as we will describe during this learning activity. We have got another pressure which is known as trans-airway pressure. This is the pressure across the wall of the airways. So it is equivalent to the difference in pressure between the pressure inside the airways and the pressure inside the pleural sac. And it is calculated by subtracting the pleural pressure from the airway pressure. The transpulmonary pressure, this is the difference in pressure between the pressure inside the alveoli and the pressure in the pleural sac which surrounds the alveoli. And it is calculated by subtracting the pleural pressure from the alveolar pressure. And positive value, when I've got a positive value because the pleural pressure is a negative, so if we've got a positive value, it indicates that the pleural pressure is less than the alveolar pressure. So here we have the atmospheric pressure, the barometric pressure, which is zero pressure or 760 millimeters of mercury. Here we have got the alveolar pressure, which is also known as intra-alveolar pressure or pulmonary pressure, and this is the intrapleural pressure or the pleural pressure. Now, the difference in pressures between the alveolar pressure and the intrapleural pressure is what we call the transpulmonary pressure. And this transpulmonary pressure, it is the driving force for lung expansion. It is the pressure which is responsible for expansion of the alveoli. If the alveolar pressure is higher than the intrapleural pressure, then the alveoli will expand. And the opposite is true. If the pleural pressure is higher than the alveolar pressure, then the alveoli will collapse. Now, the difference in pressure between the atmosphere, the atmospheric pressure, and the alveolar pressure, this is the driving force for the flow of air into and out of the lungs. When the alveolar pressure is less than the atmospheric pressure, air gets in. And when the alveolar pressure higher than the atmospheric pressure, air gets out from the alveoli into the atmosphere. So airflow is directly proportional to the pressure gradient between the atmospheric pressure, which is constant as I've just mentioned, and the alveolar pressure, and is inversely proportional to the resistance of the airways to the flow of air. So then what determines this pressure gradient? Since the atmospheric pressure is constant, so changes in the alveolar pressure are important for the generation of this pressure gradient. So what then determines the alveolar pressure? What determines how much pressure do we have inside the lungs, inside the alveoli? Based in Boyle's law, the alveolar pressure is determined by the volume of the lungs, the volume of the alveoli, and the number of molecules or the number of moles of air inside the alveoli. So at the end of normal resting eupneic breathing at the sea level, the barometric pressure and the alveolar pressure are both equal and equivalent to 760 millimeters of mercury or 1000 centimeters of water at the sea level, and they are both designated values of zero. Remember, their values are not zero, we just call them zero. So since they are both equal, so there is no pressure gradient then between the atmosphere and the alveoli at the end of normal resting eupneic breathing at the sea level, and there is no movement of air. And we need to remember that the barometric pressure is constant throughout the entire breathing cycle. It doesn't change. What about the intrapleural pressure? We've just mentioned that at the end of normal resting eupneic breathing, the intrapleural pressure is 5 millimeters of mercury or 5 centimeters of water less than the atmospheric pressure, and is therefore designated a value of -5. The transpulmonary pressure, the PL, is a positive pressure, is positive 5 centimeters of water or positive 5 millimeters of mercury, because the pressure inside the alveoli is atmospheric pressure, which is 0, while the pressure inside the pleural sac is 5 centimeter of water or 5 millimeter of mercury lower than the atmospheric pressure. And remember, the transpulmonary pressure is the pressure difference between the alveolar pressure and the pleural pressure, and since it is a positive, it indicates that the pressure inside the alveoli is 5 millimeters of mercury or 5 centimeters of water higher than the pressure inside the pleural sac. So this helps the alveoli to keep them expanded and prevent their collapse. Now let's then describe the mechanics, the dynamics of the breathing cycle in details. What happens during inspiration, which is also known as inhalation? The first step is that we've got a neural input from the respiratory center in the medulla oblongata. So we have action potentials traveling down from the medulla oblongata through the spinal cord and reaching the muscles of inspiration, reaching the inspiratory muscles, the diaphragm and the external intercostal muscles. The muscles respond by contraction, so the diaphragm contracts and the chest cavity increases in the vertical dimension, and the external intercostals contract, and this increases the chest cavity in the transverse dimension and in the anteroposterior dimension, because the contraction of the external intercostal muscles lift the chest cage upwards and outwards. So this results then into expansion of the chest wall. Remember, air has not gone in yet. The chest wall expands because of contraction of the muscles of inspiration. Now, as the chest wall expands, because the pleura is attached to the chest wall, this pulls on the intrapleural fluid. So the diaphragm, when it moves down, it moves along with it the parietal pleura. And when the chest wall moves outwards and upwards, it moves along with it the parietal pleura. So based on Boyle's law, then what happens to the volume of the pleural sac? The volume of the pleural sac increases, and because of the increase in the volume, the pressure inside the pleural sac becomes even less. So now the transpulmonary pressure, which was 5, just 5 at the end of normal resting expiration, is now something like, say, positive 8, because the intrapleural pressure is now negative 8. So when it is negative 8, so it means that the alveolar pressure is 8 millimeters of mercury or 8 centimeters of water higher than the pleural pressure. So what happens to the alveoli as a result of this? The pleural sac, which is surrounding the alveoli, is becoming more negative. So the alveoli expand as a result of this, because the transpulmonary pressure, which is the driving force for expansion, has increased from positive 5 millimeters of mercury to positive 8 millimeters of mercury, or from positive 5 centimeters of water to positive 8 centimeters of water. So alveoli expand because the transpulmonary pressure has increased, and it is the driving force for alveolar expansion. This increases the volume of the alveoli. Remember, air has not gone yet. And when the volume of the alveoli increases, the pressure inside the alveoli decreases. So now the intra-alveolar pressure is less than the atmospheric pressure, and air gets in. So and the increase in the transpulmonary pressure from positive 5 millimeter of mercury to positive 8 millimeters of mercury, or from positive 5 centimeter of water to positive 8 centimeter of water, increases the lung volume. Intra-alveolar pressure decreases. Now the atmospheric pressure becomes higher than the intra-alveolar pressure, and we have got a pressure gradient between the atmospheric pressure and the alveolar pressure, and air flows in. And as air flows in, the number of molecules of air inside the alveoli increase, and this elevates the alveolar pressure and bringing it back equal to the atmospheric pressure by the end of normal resting inspiration or inhalation. So during then inspiration for eupneic breathing at the sea level, the muscles of inspiration contract first. This increases the thoracic cavity volume in all dimensions and pulls the pleural sac as well, increasing the pleural sac volume. So the pressure inside the pleural sac decreases and becomes negative 8. So it is now 992 centimeter of water, causing the alveoli to inflate because of the increase in the transpulmonary pressure. If the alveolar inflation increases the volume, and this decreases the pressure inside the alveoli. The pressure inside the alveoli becomes negative 1 millimeter of mercury or negative 1 centimeter of water, which is equivalent to 999 centimeter of water. Now, the atmospheric pressure is 1000 centimeter of water, and the alveolar pressure is 999 centimeter of water. So there is a pressure difference. I understand that it is only 1 centimeter of water or 1 millimeter of mercury, the difference in pressure between the atmospheric pressure and the alveolar pressure, but this 1 millimeter of mercury or 1 centimeter of water difference in pressure is enough to drive air in from the atmosphere into the alveoli. So air moves into the alveoli down this 1 millimeter of mercury or 1 centimeter of water pressure gradient. So air fills the alveoli, increasing the number of air molecules and increasing the alveolar pressure back towards 0. So, and when the alveolar pressure now is 0, is 1000 centimeter of water, air flows stops because now the pressure gradient is no more existing. So by the end of then normal eupneic inspiration at the sea level, the transpulmonary pressure is approaching positive 8 millimeter of mercury or 8 centimeter of water. So this is again the story which we need to understand well, because some people think that the chest expands during inspiration because air gets in, and this is not correct. In fact, the chest expands first, and it is because of expansion of the chest which lowers the pressure inside the alveoli, air gets in. So at the end of inspiration for eupneic breathing at the sea level, thoracic cavity has stopped expanding. So thoracic cavity is at its greatest volume because now 0.5 liter of air or 100 milliliters of air have entered into the alveoli from the atmosphere. The intrapleural pressure is negative 8 millimeter of mercury or negative 8 centimeter of water, which is actually 992 centimeter of water, which is the lowest level during breathing. And the alveoli, they have stopped expanding. Why? Because the alveoli are at their greatest volume, and the pressure inside the alveoli is 0, which is 1000 centimeter of water or 760 millimeter of mercury due to air filling into the alveoli. So now the alveolar pressure is now equal to the barometric pressure, and the pressure gradient is no more existing between the atmosphere and the alveoli, and no air flow. Air stops flowing into the alveoli, and the transpulmonary pressure is positive 8 centimeter of water, which means that the pressure inside the alveoli, which is 0 by the end of normal resting eupneic inspiration, which is 1000 centimeter of water, is higher, 8 centimeter of water higher than the intrapleural pressure, which is 992 centimeter of water. And at this level, inspiration ends. So the lungs, now the alveoli, have been inflated with 0.5 liter of air or 100 milliliter of air by the end of inspiration for eupneic breathing at the sea level. So now inspiration ends. What happens next? Expiration. So what happens then during expiration for eupneic breathing at the sea level? Now the muscles of inspiration relax, and the relaxation of the diaphragm and the external intercostal muscles, they bring back the thoracic cavity to its original volume, and the pleura moves with the thoracic cage, so this decreases the pleural space volume. So now the pleural space volume returns back and approaches its resting levels. So the pleural pressure now increases towards negative 5 centimeter of water or negative 5 millimeter of mercury, which is actually 995 centimeter of water or 755 millimeter of mercury. So now the transpulmonary pressure is decreased, so this decreases the alveolar volume towards the resting, which is the end of expiration level. So now, because of the decrease in the volume of the alveoli, the alveolar pressure becomes higher than the barometric pressure and is now positive 1. Positive 1 means 1 millimeter of mercury or 1 centimeter of water higher than the atmospheric pressure. So now the pressure inside the alveoli is 1 millimeter of mercury or 1 centimeter of water higher than the atmospheric pressure, and this pressure gradient is enough for air to rush out of the lungs down this pressure gradient, and air keeps on moving from the alveoli to the atmosphere, and this decreases the molecules of air inside the alveoli, so this decreases the pressure inside the alveoli, and when the pressure is now back to the atmospheric pressure, which is 1000 centimeter of water, expiration ends. And by the end of expiration, the normal resting expiration, the sea level, the transpulmonary pressure approaches its resting value, which is positive 5 millimeters of mercury or positive 5 centimeter of water. So this is expiration. So the diaphragm and the inspiratory intercostal muscles, the external intercostal muscles, stop contracting and they relax. So the chest wall recoils inwards. The intrapleural pressure moves back towards its non-inspiratory value. Transpulmonary pressure moves back towards its pre-inspiration value. The recoil of the lungs also happens towards its pre-inspiration size, and air in the alveoli becomes compressed. So this increases the intra-alveolar pressure, which now becomes greater than the atmospheric pressure, and now the pressure gradient of 1 millimeter of mercury or 1 centimeter of water is established between the alveoli and the atmosphere, which is enough for air to flow out of the lungs. So this is again the story or the dynamics of the breathing cycle, inspiration and expiration. So these are our alveoli, and these are the molecules of air inside the alveoli. So this is the position at the end of normal resting eubnic expiration at the sea level. So this is the intra-alveolar pressure is 0, the atmospheric pressure is 0, so there is no movement of air. And we have got this number of moles or molecules of air inside the alveoli. So this is the alveolar pressure is 0, and there is no movement of air. Now with inspiration, what happens first? The alveoli expand because of the increase in the transpulmonary pressure, because of the movement of the chest wall pulling the pleura, and the molecules of air inside the alveoli hasn't changed yet. So because of the increase of the volume, the pressure inside the alveoli decreases to -1 millimeter of mercury, -1 centimeter of water. So now a pressure gradient is established between the atmosphere and the alveoli, and air gets in. So now more moles or molecules of air move in and keep on moving till the pressure is back to 0. So how much air gets in during normal resting eubnic inspiration at the sea level? Approximately 0.5 liters of air, or 500 milliliters of air. And when this happens, now the pressure inside the alveoli by the end of the normal resting eubnic inspiration is atmospheric pressure, and the diaphragm and the external intercostal muscles, they relax, and the process of expiration begins when the chest cavity returns back to its original size. And when this happens, the alveoli are compressed, and this elevates the pressure inside the alveoli to +1 millimeter of mercury or centimeter of water, and thus air gets out. How much air gets out during normal resting eubnic expiration at the sea level? The same volume of air which gets in, which is approximately 0.5 liters or 500 milliliters of air. And when this happens, the number of molecules or moles of air inside the alveoli has decreased, and then the alveolar pressure is back to the atmospheric pressure, and the process of expiration ends. So this is again a summary of the dynamics of the normal breathing cycle, the normal eubnic inspiration and expiration at the sea level. And this figure summarizes the changes in the pleural pressure, the alveolar pressure, and airflow, and the volume of air which gets in and out during the normal breathing cycle. So this is the initial position. This is the position at the end of normal resting eubnic expiration. On the x-axis we have the time, and on the y-axis we have different parameters. So in this part of the figure we have the intrapleural pressure measured in centimeter of water. So at the end of normal resting eubnic expiration it is -5 centimeter of water, and during during normal resting eubnic inspiration it reaches to -8 millimeter of mercury or -8 centimeter of water, and during expiration it returns back to its pre-inspiration level. This is the alveolar pressure measured in centimeter of water. This is the position at the end of normal resting eubnic expiration. It is atmospheric 0. Then it is reduced to -1 centimeter of water, or -1 millimeter of -1 millimeter of mercury, -1 centimeter of water, and air gets in, and because air gets in, the pressure increases back to atmospheric 0, then pressure increases to +1, and it is because of that air gets out, and as air gets out, so the pressure reduces back to 0. So this is the volume of air which gets in and out. So approximately 0.5 liters of air gets in during inspiration, and the same volume of air gets out during expiration, and this volume we call it the tidal volume. So now let's describe a very important pathophysiological condition related to the breathing cycle, which is pneumothorax. So what is pneumo? Pneumo means air. So pneumothorax means the presence of excess air in the pleural sac, and when air accumulates in the pleural sac, this elevates the intrapleural pressure, which is now no longer less than the atmospheric pressure. So now the pressure inside the pleural sac is higher than the pressure inside the alveoli. So this causes alveolar collapse. Remember, we do not have air inside the pleural sac. So what could be then the reason for air entering into the pleural sac? What could be the causes of pneumothorax? It could be due to trauma, and this trauma could be a penetrating bullet wound or knife stab wound, or non-penetrating injury from a broken rib. So now air could get into the pleural sac from the outside environment, or air could get into the pleural sac from the chest cavity itself. Another cause for pneumothorax is positive pressure ventilation, and this will be described to you later on. Or it could be spontaneous, something which happens spontaneously. And spontaneous pneumothorax, it could be idiopathic. Idiopathic means primary. We don't know the cause of it, and it could be due to rupture of a lung cyst, and this usually happens in thin and tall men. Or it could be secondary due to lung diseases, like a disease known as emphysema, and in this emphysema there are blips of air forming on the visceral pleura, and when this rupture, this results in escape of air from the lungs, and this air fills the pleural sac, elevating the pressure inside the pleural sac, resulting into lung collapse. So this is what we have here. This is now a trauma. Air gets into the pleural sac, elevating the pressure inside the pleural sac. So now the transpulmonary pressure is no more positive pressure, rather now the pressure inside the pleural sac is higher than the alveolar pressure, resulting into collapse of the alveolar lung collapse. Same thing here. This is a trauma, penetrating chest wall injury because of bullet injury, or it could be non-penetrating because of a broken rib. So now air gets into the pleural sac, causing pneumothorax. Intrapleural pressure becomes higher than intra-alveolar pressure, causing lung collapse. So in pneumothorax, the intrapleural pressure is no longer negative, and lung collapses because of its inwardly directed recoil force. Pneumothorax could be simple pneumothorax or tension pneumothorax. Simple pneumothorax means that air can enter and exit the pleural sac, so it equilibrates, and this causes equilibration of either the alveolar pressure and the pleural or the barometric pressure, and So now the pleural pressure is equivalent to the alveolar pressure, or the barometric pressure is equivalent to the pleural pressure. So this is a chest x-ray of left-sided pneumothorax. So air now has accumulated inside the left pleural sac, resulting into the collapse of the left lung. And the reason for this simple pneumothorax, it was a broken rib. The other type of pneumothorax is known as tension pneumothorax, and this is much more serious than simple pneumothorax because air enters the pleural sac, it enters the chest cavity, but cannot escape. And it has been postulated that the reason why air enters into the pleural sac and cannot escape from the pleural sac in tension pneumothorax, that a valvular mechanism might have developed through which air can be sucked into the pleural sac during inspiration, but air cannot be expelled during expiration. So what happens here now, the enteral pleural pressure keeps on increasing and it remains positive throughout the breathing cycle. Remember, in simple pneumothorax, the enteral pleural pressure is equivalent to the alveolar pressure, or the pleural pressure is equivalent to the barometric pressure. But in tension pneumothorax, the enteral pleural pressure remains positive throughout the breathing, and the lung deflates further. So this is the lung deflates further, and it might even push the mediastinum and compresses the heart and the great blood vessels. So this might reduce the venous return to the heart, and it is a medical emergency, so we need to insert a needle in order to decompress the chest. And this is, can you see here the trachea? This is the thoracic vertebra, and this is the trachea, air in the trachea, and the trachea is in the mediastinum, and it has pushed to the left side as a result of left-sided tension pneumothorax. So the left-sided pleural sac is full of air and air cannot get out. We have finished with describing the dynamics of the breathing cycle and the pathophysiology of pneumothorax. And now let's describe another important topic related to pulmonary ventilation, which is spirometry. What is spirometry? Spirometry is the study of the lung volumes and lung air flow rate using an instrument which is known as the spirometer, which measures the lung air volumes and the lung air flow rates. This is this old spirometer, and now we have got the more recent digital spirometers. So the spirometer is essentially an instrument which measures the lung air volumes and lung air flow rate. So far we have known only one lung volume, which is the tidal volume, and we've mentioned that the tidal volume is the volume of air which gets in or gets out during the normal resting eupneic breathing cycle at the sea level. So this is the old spirometer, where we have a rotating drum and we have got here a needle. So when air gets in, the needle moves up, and when air gets out, the needle moves down, recording the lung volumes. So are there other lung volumes apart from the tidal volume? Yes, there are other lung volumes. So now on the x-axis, if we have the time, and on the y-axis we have the lung volumes. So this position is the end of normal resting expiration. Do we have air inside the two lungs by the end of normal resting yupnik expiration? Yes, we do have. We do have this much of air inside the two lungs. So from this position, which is the position of the end of normal resting yupnik expiration, if someone takes normal inspiration, then a tidal volume gets in. So this is the position at the end of normal resting yupnik inspiration at the sea level. Then this is expiration. So this is the position at the end of normal resting yupnik expiration at the sea level. Then another inspiration. This is the position at the end of normal resting inspiration at the sea level. Now, if from this position, which is the position at the end of normal resting yupnik inspiration at the sea level, you ask this subject to breathe out, to expire with force, how much air gets out? Now, the air which gets out during forced expiration includes the tidal volume, and this subject can expire an additional volume of air which is known as the expiratory reserve volume. So the expiratory reserve volume, this is the volume of air which can be expired further using forced expiration. And even when someone has already expired the tidal volume and the expiratory reserve volume, this doesn't mean that his two lungs are empty and have no more air. Still, the two lungs have got a volume of air which cannot be expired even with forced expiration, and this volume of air is known as the residual volume. And definitely we cannot then measure the residual volume using the spirometer, because this is the volume of air which remains inside the two lungs even after forced expiration. Okay, so again, this is the position of the end of the normal resting yupnik expiration at the sea level. If from this position you ask someone to take forced inspiration, to inhale with force, how much volume of air gets in? The volume of air which gets in includes the tidal volume, of course, and an additional volume can get in, which is known as the inspiratory reserve volume. So the inspiratory reserve volume is then the volume of air which can be inhaled in addition to the tidal volume using forced inspiration. So what is this position then? This is the position at the end of forced inspiration. We have then lung volumes, each of which is composed of only one volume, like the residual volume, and this is the volume of air which remains inside the two lungs even after forced expiration. And we have the expiratory reserve volume. This is the volume of air which can be expired further after the tidal volume using forced expiration. And we have got the tidal volume. This is the volume of air which gets in and gets out during normal resting yupnik breathing. And we have the inspiratory reserve volume. This is the volume of air which can be additionally inhaled after the tidal volume using forced inspiration. So these are the lung volumes, each of which is composed of only one volume. We have got other lung volumes which are composed of two or more lung volumes. For example, we have a lung volume known as functional residual capacity. So the lung volumes, which are composed of two or more lung volumes combined together, are known as capacities. The functional residual capacity, this is the functional residual capacity, and it is abbreviated as functional residual capacity. It is composed of what? The functional residual capacity, the expiratory reserve volume and the residual volume. So this much air. So what is then the functional residual capacity? The functional residual capacity is the volume of air which remains inside the two lungs at the end of normal resting eupneic expiration at the sea level. And it is composed of the expiratory reserve volume plus the residual volume. And since we cannot measure the residual volume using the spirometer, we cannot also measure the functional residual capacity using the spirometer, because the residual volume is a volume in the functional residual capacity. Another capacity is the inspiratory capacity. It is composed of the tidal volume plus the inspiratory reserve volume. We call it inspiratory capacity. So this is the volume, the inspiratory capacity. This is the inspiratory capacity. It is the volume of air which can be inspired from the end of normal resting eupneic expiration using forced inspiration. So it is composed of tidal volume plus the inspiratory reserve volume. Another capacity is what we call the vital capacity. And this vital capacity, this is the maximal volume of air which can be inspired or expired using the maximal inspiratory and expiratory effort. So it is composed of the tidal volume plus the inspiratory reserve volume plus the expiratory reserve volume. Another lung capacity is known as the total lung capacity. So what's this total lung capacity? This is the total amount of air which we can have inside the two lungs after forced inspiration. So it is composed of the inspiratory reserve volume, the tidal volume, expiratory reserve volume, and residual volume. Can we measure the total lung capacity using this spirometer? We cannot. Why? Because it also includes the residual volume. Any capacity which includes the residual volume, we cannot measure it using the spirometer, since we cannot measure the residual volume itself using the spirometer. So here we have again, this is the position at the end of normal resting eupneic expiration at the sea level. How much air do we have inside the two lungs? This is what we call the functional residual capacity. How much is the functional residual capacity? Approximately 2,300 milliliters of air still remaining inside the two lungs by the end of normal resting eupneic expiration at the sea level. So now when we breathe in quietly during eupneic inspiration at the sea level, an additional 0.5 liters of air, 500 milliliters of air are added to these 2,300 liters of air, and now the volume is what? 2,800, because the tidal volume was added to the functional residual capacity. So now another, this is the tidal volume expired, bringing back the volume to the functional residual capacity, 2,300. This is a tidal volume inspired in. Now the volume is 2,800. Now if from this position, which is the position at the end of normal resting eupneic inspiration at the sea level, you ask this subject to breathe out with force, he's going to breathe out these 500 milliliters of air from 2,800 to 2,300 which are the tidal volume, then additional 1,100 milliliters of air, which is the expiratory reserve volume, and then the remaining, which is 1,200 milliliters of air, which cannot be expired even using forced expiration, is what we call the residual volume. So now this is the position at the end of normal resting eupneic expiration. If you ask someone to breathe in with force, he's going to breathe in first the tidal volume, 500 milliliters of air, then additional 3,000 milliliters of air, which we call the inspiratory reserve volume. Now this is the position at the end of forced inspiration. If from this position you ask someone to breathe out with force, he's going to breathe out the inspiratory reserve volume, the tidal volume, and the expiratory reserve volume. And this is what you call the vital capacity, which is equivalent to the 3,000 inspiratory reserve volume plus 500 the tidal volume plus 1,100 the expiratory reserve volume. So it is approximately 4,600 milliliters of air. Now this much air, which is the residual volume, 1,200 milliliters of air, cannot be expired even using forced expiration. So this table summarizes the definitions of the lung volumes and lung capacities, and we've mentioned these in the previous slides. So the tidal volume, again, this is the volume of air inspired or expired during eupneic breathing, is approximately equivalent to 500 milliliters of air. Inspiratory reserve volume, approximately 3,000 to 3,300 milliliters of air, the range. And this is the maximum amount of air that can be inspired at the end of normal inspiration. Expiratory reserve volume, approximately 1,000 to 1,100 or 1,200 milliliters. And this is the maximum amount of air that can be expired at the end of a normal expiration. Residual volume, we cannot measure it using the spirometer because this is the volume of air that is trapped inside the lungs even at the end of normal the forced expiration. Now the above values are for an average size healthy adult male. Adult females, they would have 20% smaller volumes, and the above volumes are also dictated, they are determined by the sex, by the age of the subject, by its height, by the fitness, and his health status. So these are the volumes, and again these volumes, each volume is composed of a single volume, and these are the capacities. Functional residual capacity is equivalent to the expiratory reserve volume plus the residual volume. The vital capacity is equivalent to the inspiratory reserve volume plus tidal volume plus the expiratory reserve volume. The total lung capacity includes all volumes, so it is equivalent to inspiratory reserve volume plus tidal volume plus expiratory reserve volume plus the residual volume. So these are the capacities. Total lung capacity approximately 5,700 up to 6,200. Vital capacity is the maximal volume of air that can be expired from the lungs after a maximal inspiration. This is the inspiratory capacity, functional residual capacity. So this figure again summarizes, these are the individual volumes. This is the tidal volume, which is inhaled and exhaled during eupneic breathing at the sea level. This is the extra additional volume which can be inhaled, the inspiratory reserve volume. This is the additional volume which can be exhaled, the expiratory reserve volume. And this is the volume which cannot be exhaled using forced expiration, which is the residual volume. These are the capacities. This is the functional residual capacity, and this is the inspiratory capacity. This is the vital capacity, and this is the volume of air which can be expired with force from the position of forced inspiration. So when someone is asked to expire with force from this position, which is the position at the end of forced inspiration, he's going to expire the inspiratory reserve volume, the vital capacity, and the expiratory reserve volume, and he cannot expire the residual volume. And this is the total lung capacity. Now, since we cannot measure the functional residual capacity using the spirometer, we need to use another way for measuring the functional residual capacity, and the method which we use to measure the functional residual capacity is known as the helium dilution method. So for the helium dilution method which we use to measure the functional residual capacity, we use the concept that if we have a quantity of material which is distributed over a particular volume, then the concentration of the material will be C1 and the volume will be V1, and the quantity of the material will be equivalent to the concentration of the material multiplied by its volume of distribution. Now, if the volume through which this material has distributed has increased and now becoming V1 plus V2, and the quantity remains the same, then the concentration will obviously be decreased and becomes C2. Now, since the quantity of the material hasn't changed, what has changed is the volume. The volume has increased and the concentration has decreased. Then the quantity after the addition of V2 will be the same, but will be now C2, which is the new concentration, multiplied by the two volumes, the original volume and the additional volume. Thus, the quantity here is the same, so C1 multiplied by V1 is equivalent to the same quantity, which is C2 multiplied by V1 plus V2. So C1 multiplied by V1 is equivalent to C2 multiplied by V1 plus C2 multiplied by V2. We can have this in one side of the equation, so this goes with the negative side here. So C2 multiplied by V2 is equivalent to C1 multiplied by V1 minus C2 multiplied by V1, and we can calculate V2, which is the additional volume through which this substance has been distributed. So V2, this is the additional volume, is equivalent to C1 multiplied by V1 minus C2 multiplied by V1 divided by C2. So we can use the same concept now to measure the functional residual capacity. What is this material which is distributed? The material is helium. What volumes? The volume of the spirometer, the amount of air which is in the spirometer, and the additional volume is the functional residual capacity. So V2 is our functional residual capacity. So here we have air. So now we ask this subject to breathe out quietly. So now at the end of normal resting eubnic expiration at the sea level, the amount of air inside the two lungs is equivalent to the functional residual capacity, as we mentioned in the previous slides. Now this subject breathes known concentration of helium, which is C1, in a known volume, which is V1. So this is C1 and this is V1. This is the known amount of helium which was originally present in the spirometer. Now we ask this subject to breathe in. So now helium is going to be distributed over an additional volume, which is V2, which represents the functional residual capacity. And we can use the same concept now. V2, which is the functional, this is after equilibration. V2 is the functional residual capacity, is equivalent to V1 multiplied by C1 minus C2 divided by C2. The same concept here. V2, which is the functional residual capacity, is equivalent to C1 multiplied by V1 minus C2 multiplied by V1 divided by C2, and we can then measure V2, which is our functional residual capacity after helium equilibrates in the volume of the spirometer and the functional residual capacity. And that's the end of these learning activities on pulmonary ventilation. And here we have the intended learning outcomes again, and thank you very much for listening. And again, you need to listen to this video recording before coming to the lecture.